Hey everyone, thanks so much for joining me. I'm your host, Jared Taylor. Joining me today is the CEO and the Chief Scientific Officer of Ratio Therapeutics. Really excited to have you gentlemen on the podcast today. How are you?
Great. Happy to be here. Nice to be here, Jared.
Well, I'd love to start by giving you each an opportunity to, talk about yourself, like give us the snapshot of each of you, and then let's dive more into Ratio Therapeutics. But whatever order you gentlemen would like to go into, tell us a little bit about yourself.
Great. I'll kick off and then I'll pass it to John to kind of give the how we got here, his background and how we got here. So Jack Hoppin, I'm a mathematician by training with a background in molecular imaging. Have worked in the nuclear imaging and nuclear medicine space basically my whole career, post grad school, postdoc, and on. Relevant to this conversation, started in the imaging hardware business, making an imaging system. I was in a German lab, moved to Hungary where we made that into a product. A few years in, thought it would be a good idea to really focus on the software and services aspect of that technology, and started a company in Boston in 2008 by the name of Invicro. Grew that company over the course of a decade or so, and to a few hundred people, doing a lot of imaging research in pharmaceutical trials, both discovery all the way through to late stage trials, and very technical company, and sold that company to Konica Minolta Precision Medicine in 2017. Stayed on at Konica Minolta, first continuing to run Invicro, and then as the president of their precision medicine business, which had imaging, genomics, and digital pathology. That's along the way. I met John Babich a couple times, but we reconnected in 2018. In 2020, I decided to retire, which I failed at, as you're about to hear, and I'll pass it to John.
I would say he failed up.
Yeah, that's it. Yeah, exactly.
So John Babich, my background's in pharmaceutical chemistry. My whole career has been devoted to nuclear medicine. Started out at Brookhaven National Laboratories, did some work at NASA and the Johnson Space Center, looking at applications of imaging and how it affects in how zero G affects physiology. Moved to the UK where I did my PhD at the Institute of Cancer Research. Went from there to MGH and really was focused on the study of drugs and their distribution. Worked with several big pharma companies on trying to understand how their drugs were working, both throughout the body, but particularly in the brain. Got an interesting itch to start a company back in 1998, a company called Molecular Insight Pharmaceuticals. Took that out and developed that company in Cambridge, Mass. Built that to about 120 people at a peak. Took that public in 2007. Had a series of really cutting-edge clinical programs, including prostate cancer and neuroendocrine cancer, things that are top of the mindset of the topic that we'll talk about today. And sold that company to Progenics in 2013. Took a diversion back into academia and was thinking about really how do we really think about targeted radiotherapeutics in general. And we'll get into that in a second, but the idea of actually being able to inject a radioactive drug to find and treat metastases of cancer anywhere in the body. And as we play with that idea and fine tune that idea, we came up with a very interesting scaffold, a very interesting platform where we can tune the pharmacokinetics in the body. And that led to some very interesting thinking in that, which is, can we optimize the PK? Is there like a golden rule about what's required for optimal localization in cancers? And at that time, I reached out to Jack. I knew Jack by his background and him and his work in the field. I said, Jack, I have an interesting problem. I think that your mathematical expertise I'd love you to think about this problem with me. And that was, how do we optimize the delivery of drugs, radioactive drugs, to tumors? And we got reacquainted back in 2018. And we were working on a program in prostate cancer at the time. We had spun a company out of my lab at Cornell called Ratio Therapeutics that eventually got acquired by Bayer Pharmaceuticals. During that time, Jack was on the board. We really worked hard to get that deal done together. And then at the actual execution of that deal, or a week before, it became apparent to us that we needed to start another company. So we started a company essentially with the same technology. We sold a particular use to Bayer in prostate cancer, but we recapitulated all those licenses and started Ratio Therapeutics. We wound up with a very good relationship with Bayer out of that, and also another company called Lantheus Pharmaceuticals here in Massachusetts, and bootstrap through those relationships on our own internal investments, and created Ratio Therapeutics back in June of 2021. And here we are today. We've got our our first IND. We've got clinical programs going on, a lot of exciting things happening, and we're opening a headquarters, probably at the end of February here in the Seaport area of Boston. So it's kind of a short story.
In an absolutely beautiful area too. You have to throw that in there, because I know the building you're talking about, and that area with the marina and everything. I mean, you guys have a stressful day today, right? Being able to go outside real quick on a call or something and see that marina, that sun when it's not winter, right? Will be great for the team. Thanks so much, John. Jack, I want to touch on something that John mentioned, right? He mentioned, you being a mathematician, which is super interesting. I want to dive more into like what you bring to Ratio as a mathematician. Obviously, you're more than just a mathematician, but I want to focus in on that piece, because I don't hear that as much, and I really want to focus in on it.
Sure. So John really alluded to it that by definition, this class of therapeutics is very quantitative. We can image the drug and then we treat with the drug. And John is a very multimodal scientist who knows really had thought of the problem holistically. And when he came with this notion of tunable pharmacokinetics, which is tunable biodistribution strategy for the drugs, my background is really in studying and quantifying the biodistribution of drugs. And there's a lot of math in it. There's a lot of math in imaging itself. And in this case, there's very quantitative measures of if you are going to be able to create a drug that's going to be effective. And when John showed up at our door and we met, I really thought the way he looked at the problem, which is how do we optimize this delivery system? How do we optimize the ratio of residence to the tumor, to the ratio to the off target? That's really what this activity is about. And so we as a group, we're only 22 people right now, but very technical group across a broad spectrum of disciplines. And math is one of the components, math, physics, chemistry, biology, that supports the pharmacology to understand the pharmacodynamics of the drug. And we have a core nucleus here that's really strong. I cannot help but to bring up that when we think about one of the advantages of this class of drugs, we're always going to image those drugs. We do nuclear tomography, usually 2 modalities, sometimes 3. So we have emission tomography, SPECT and PET, where we inject the radioactivity and image where it goes. And then we have transmission tomography, where we're using an X-ray source and looking at the attenuation map that is our bodies, the bone, all the different organ attenuation maps. That tomography is the writing of slices, so I think it's only appropriate that you have tomographers on slice of healthcare. Shameless pitch for slicing. So that stacked slice of images really gives us the best biomarker readout for our drugs possible, and then we go to an alternative where we're trying to irradiate the tumor by injecting, radionuclides on a drug that we're trying to target the tumor at. So that's the team, and so I think it's really, math is just one pillar of what comes together here.
Now, the company owns 2 R&D platforms, right? And if I butcher the second one, please forgive me. But Trillium and Macropa, did I say it right?
Perfect. We accept all pronunciations of that. That's totally
perfect. So that's really cool. Can you talk a little bit about how each of them work?
Sure. The Trillium is the platform that I alluded to in terms of the trying to understand how you can design a molecule and make it as, I'll try to make it as simple as possible. So Trillium is a flower, right? So it's a three-pointed, three-petal flower, and it sounds better than trifunctional scaffold. So we have this Trillium scaffold, and it has three components to it, right? It has a component to it that allows the binding to a protein that's on the surface of a cancer. So that's a tumor targeting component. And then it has a component that takes the radionuclide with it, right? So what we're doing is really using the radionuclide, as Jack alluded to. We can see that, we can image that, but we want to swap out the isotope for a therapeutic benefit to the patient. So it carries a payload. And then the third piece to this is this PK modulation motif. And what we have is a series of compounds that bind albumin reversibly. So they'll bind, lots of drugs, many drugs bind albumin, right? It's part of the solubilization after you take a tablet. Things wind up, things like ibuprofen and other things, warfarin, they bind to proteins in your plasma, and that's how they get delivered to the body and how they get solubilized. But you can actually use it as a sort of feature to the molecule where you can dial in how much binding you have, and then it changes the plasma curve. So and everything we do is injectable. So as soon as you inject the drug, it's going to see albumin, and it's going to be circulating around the body as a function of how tightly it binds albumin. So we want something to stay there for a long period of time, to have multiple passes through the tumor bed. Then we can put something that's fairly strong, and that gives us the ability to do that. If we want to do something, say, for imaging, where we don't really need multiple passes, we want a quick flush, and then we want everything to escape the plasma, then we have a very weak albumin binder on there. So we have this range of affinities or strengths that we can dial in, and that's the Trillium scaffold. We have the ability to change the payload, change the albumin binding domain, and actually change the tumor targeting domain. And we've been successful with two clinically scaffolds that way. The second scaffold really is a unique way of binding an alpha emitter called actinium-225. So this is a very potent anti-cancer alpha emitter. But the chemistry is kind of bulky, right? This is something found in the bottom of the periodic table. It's big, it's clunky, it's not something you have a lot of experience with collectively. But it turns out that Macropa actually binds this thing like hand in a glove, right? It just goes in there very quickly. It will not come out in any reasonable, under any reasonable circumstances, both in a vial or in a patient or an animal subject. So we have the way of actually putting these very potent isotopes onto our molecules very, very quickly and robustly, and it makes manufacturing much simpler and allows us to use actinium in a variety of applications. And not just for Trillium, but we can also use it for other constructs, right? Antibody constructs, small molecules, peptides, whatever it is. It allows us to basically attach one of those molecules onto any vector and allow us to label with actinium and create a therapeutic out of it. So it's quite unique that way.
And by the way, guys, I would love to have you both back on at some point, like on a panel, to talk about this with some of your peers. Maybe you alluded to a good relationship that you have. I think it was it Bayer? Was that what?
Correct.
Maybe we could have someone from Bayer come on as well to really dive into more of what you're talking about. This is your first time on Slice of Healthcare, so I appreciate you giving us a really good overview of everything that you're doing. I want to quickly shift focus to before the conversation we were talking about JPM a little bit. I did not go this year, but I know John, you did. Can you tell us a little bit, like how was the buzz? How was it? Did you stay dry?
Yeah, this is Jack. Yeah, I
Sorry.
I did go. No problem. I went. It was exciting to be back in person, I would say. I've seen it better attended, as you might expect, between the weather and the kind of the time, just everything going on. But it was booming. I mean, there was a lot of energy, and I have to say, it delivered. I thought it was a really good conference. There was it might have actually been it was probably a little positive. There were a few less people there. You could actually sit down in a couple places, whereas usually you can't even find a seat anywhere. But targeted radiotherapy was certainly discussed well across the panels. We did not present. We were just in attendance. I was just in attendance and there for meetings. But the consistent messaging that we'll repeat right now is this new emerging, it's not really a new idea, but this emerging class and the adoption of this class of targeted radiotherapy with Novartis, Bayer, really leading the way, and a class of companies similar to ourselves trying to innovate in this space and make the next generation of this class of drug, covering all the reasons it's such a constructive therapy, very little side effect, works well with targeted, with immunotherapeutics. It's a very rational alternative to external beam radiation, which is used basically in half of patients. And so there was a lot of buzz. I would say from where I sat, there was a lot of buzz around targeted radiotherapy. There was a lot of buzz around gene therapy. So not really a surprise. These are
hot topics.
Hot topics. Yeah.
What are some hot topics or some exciting things that you can share for Ratio Therapeutics as we head into 2023 and beyond that you can share at this time?
You want to talk about it?
So our big push right now is really to explore the opportunity that there's a particular target that the field is very excited about. It's a target that is found on many cancers, many epithelial cancers, called fibroblast activate protein alpha. It sits not on the cancer cell itself, although it does show up in some cancers, but in a number of cancers, it's in the sort of tissue that supports tumor growth. So it's the cancer-associated fibroblast, the part of your body that reacts to a wound. Basically, the tumor shows up, as a wound, and then your body sort of reacts to it, and then in that reaction is actually supporting and covering it's kind of like building a nest for the cancer cells. And this is a very interesting opportunity. It's been shown to be very useful at finding cancers from an imaging perspective, and there's an opportunity to, at least, theoretically, that if you can go after that particular target with a therapeutic, you have the opportunity to actually diminish the stromal component or the protective component of the host in protecting the tumor, which actually might open up opportunity for immuno-oncology applications and also act as either an adjuvant or also as supporting therapy. As you know, most cancer therapy is multi-modality, multi-drug. So this would be a component to that. And we have some very exciting preliminary data in that space, and we think that 2023 will be a big push for us to move forward with the FAP therapeutic at Ratio Therapeutics.
Well, gentlemen, I want to thank you once again for coming on as guest. We should really get that panel put together because I think the audience will really enjoy it. I know you guys would have a ton of fun talking with your colleagues over at Bayer. But really appreciate you coming on here, telling your story, telling our audience your story, and then really getting them excited about what's on the horizon. And I really appreciate you talking through your two R&D programs as well, which I found really interesting. Love the names too. It's not just a brewery. Right?
Exactly. It is funny though that we are right next to the Trillium Brewery. It's like right here. So obviously there's a lot of Trillium mugs and hats and shirts around here,
Yeah, it's for the audience, when you see this where their new office is going to be too, it is literally across the street. Yeah, it's a good spot. It's really good.
That had nothing to do with it.
Yeah, nothing to do with it. Yeah. Thank you, gentlemen, so much. And I wish you all the best of luck and an exciting 2023.
Thanks so much, sir.
Appreciate it.